a quick overview of some ideas related to human development and meiosis. Now, in terms of where we are going, we are really considering how, at the initiation of life, a fertilized egg can undergo many, many rounds of mitosis to end up with a cute, adorable baby after 9 months. But additionally, why each baby born to each parental couple looks slightly different from siblings and very different from each other, despite having a lot of shared features. Typically, most babies born at 9 months have 2 eyes, 10 fingers, 10 toes, but things like their hair color, their eye color, the presence of dimples, whether they're left or right-handed, all of those unique shared features that we might see in some offspring and other offspring are the result of the type of sperm and the type of egg that come together and make a fertilized egg when consumption occurs, when fertilization occurs. So there's 3 key ideas that we need to understand before we talk about some introductory ideas related to making eggs and sperm. A, all of the genetic information that you have inherited from your parents, your paternal and maternal biological parents, were inherited, meaning you received it from them via sperm and egg, and you yourself will pass on most of that information to the next generation if you have children. Secondly, when a sperm and an egg fuse, they're combining one set of DNA that's maternal and one set of information that is paternal. We're combining 2 individual sets of DNA, of genes, of chromosomes, and combining them into a combination of the 2. Which means that after this occurs, with the exception of making sperm and eggs, all of the cells that we see undergoing mitosis to make a baby, and then how this baby grows, contains 2 copies of DNA, one from their mother and one from their father. However, additionally, all of this information will be passed on in some combination to the next generation when that child has a offspring. And that is a term that we're going to think of as sexual reproduction, the selection of genes and traits in random breeding opportunities between 2 different individuals that combine their DNA. So this isn't a cloning or a fusion type of reproductive event. So we're going to talk about a few keywords that are really important for starting to think about how we make sperm and eggs that create unique individuals. The first definition is going to be a gene. And we've talked about genes before, but genes are going to be really important as we start to think about inheritance patterns of traits. A gene is just a region of DNA. Think of it as a variety of letters that make words that codes for either a protein or a part of a protein. Now, this is important because every single gene in the human genome, we have about 30,000 genes, must be inherited when fertilization occurs. If there are genes missing when fertilization occurs, the embryo and then fetus and then the human will not be able to have sufficient development. There are genes that code for skin color, there are genes that code for eye pigmentation, there are genes that code for the proteins that allow your body to break down food. Every single one of those genes has to be present when fertilization occurs. So we can think of genes, thinking about our fat bunny ears from our mitosis unit, as just a segment of that chromosome that makes a specific protein. Now, that gene's location on a certain chromosome is what we refer to as a loci or a locus. A locus for singular, loci for plural. This is the specific region or location of a gene on a chromosome. Now, this is important because there are actually 46 chromosomes in all of our cells that are not sperm and eggs, meaning that we have 23 pairs. 1 chromosome for each pair is from a mother, and 1 of those chromosomes in a pair is from a father. Each of these contains thousands of discrete genes. Those genes have loci on certain chromosomes. For example, when we talk about males having higher prevalence of color blindness, that's because the loci for the genes that allow you to see color are found on the X chromosome. Alleles. So alleles, think of slight, subtle differences in spelling. There versus they are. There are subtle changes or variations within the gene that change the meaning of the protein ever so slightly. So a gene is a region of DNA that codes for a protein. Sometimes we have different variations of that gene. So a gene for eye color might have an allele that might produce a trait that makes a person have brown eyes or blue eyes or green eyes. They're not different genes, they're just slightly different combinations. It might be the difference between making a hamburger and you either make a smash burger or a cheeseburger. It's still a burger, you've just done subtle changes to the recipe. So when we think about a chromosome, right here, this is a single copy of a chromosome, we might have a gene with a loci that codes for eye color, and the trait is brown, so that's the allele. And we have another gene for hair color with a different loci on our chromosome, and that codes for hazel hair. We could have another individual with the same chromosome with the same genes, but the alleles might code for blue eyes and brown hair. Another thing that we could think about is just having these two chromosomes next to each other, same gene, same locus, different alleles for different types of flower colors. So allele is a variation of a gene, and the locus is the location of it. And then finally, we can talk about cells that are diploid. Cells that are diploid contain those two copies of the chromosomes. For example, humans have 23 pairs. In each pair of chromosomes, we're going to have a maternal and a paternal chromosome copy, meaning that the chromosome will have alleles for genes that are inherited from mom and from dad. So if we were talking about your parents' eye colors, my dad has green eyes and my mom has blue eyes. Those are two different alleles that I inherited in my fertilization event. Each pair obviously is made up of that maternal and paternal chromosome. Now, when we look at people's chromosomes in this regard, in terms of what's in their nucleus, we can make things that are known as karyotypes. And a karyotype is just looking at the pairing of each chromosomal pair, chromosome 1, 2, 3, 4, 5, etc. Mom's chromosome, dad's chromosome. The only difference that we tend to see in humans is the 23rd pair, which we call the sex chromosomes. Biological men tend to be XY, and biological females are almost always XX. So, some other key terms. Haploid. A haploid cell has only one copy of every chromosome. So there would only be 23 chromosomes, and there would not be pairs. There would be single chromosomes. They could be either male or female, maternal or paternal. This is what we see in our finished sperm and eggs after meiosis. They're important because haploid cells can't functionally create an organism in terms of like building something up, but their role is to fuse with another cell that's haploid to make a diploid cell. So an egg and a sperm themselves can't divide and create a human being, but once they fuse together and make a diploid cell, then we tend to see that diploid cell be able to undergo mitosis and create a full functioning human being or organism. Homologous chromosomes. Now this is going to be a term that we use to describe a pair of identical chromosomes made up of the maternal and the paternal set. Homologous chromosomes contain identical genes but different alleles depending on who your parents are and what they look like. So here are my two homologous chromosomes. For a single chromosome, I have a paternal and a maternal chromosome. Notice that after duplication, we have two X's. This is often a confusing point in meiosis, where we think that these two things are very different. Instead, they are very similar. We have just duplicated them to have two sister chromatids, or two copies of a paternal and a maternal chromosome. This is going to be really important when we create sperm or eggs in biological organisms. So a gamete is going to be the production of or the end result of our meiosis. A haploid cell, sperm or eggs, that contains one set of chromosomes. Sperm are almost always going to contain either an X or a Y sex chromosome. If a sperm contains an X chromosome and it fertilizes an egg, almost always that baby will be a female. And if it contains a Y chromosome, then that sperm fusing with an egg will make a biological male. Eggs tend to only have X chromosomes singularly in their nuclei. And then we can talk about the differences between our types of chromosomes. 22 out of the 23 pairs that I showed you on the last slide are what we call autosomal sex chromosomes, or not autosomal sex, just autosomal chromosomes. They do not determine biological sex. They might have some alleles or genes that attenuate or exasperate biological sex characteristics, but they don't determine the sex of the individual. The 23rd pair of chromosomes, X and Y, do have the majority of the roles in creating an individual having a biological sex of male or female. Those tend to be either XX or XY, but there are some other permutations that do exist. This is important because when we make our gametes, each pair is going to be made up of a maternal and a paternal chromosome, which will allow us to then create more haploid sperm and eggs in the next generation. So those are a lot of words. The gist of what I think are important is you know the difference between haploid and diploid, one or two copies of a chromosome. Homologous chromosomes are the maternal and paternal chromosomes that are identical. They're identical in the sense that they are the same size with the same genes, with the same loci of those genes, but they differ in terms of possible alleles. Alleles are variations or flavors of the gene, and the loci is the location of them on a certain chromosome. So, let's talk briefly about what's going on in meiosis. This is meiosis. We have two levels of meiosis, and you will notice when you look at this that the same actions occur. We have prophase, metaphase, anaphase, telophase. We have 1 and 2. Now we're always going to start with a diploid cell that has two copies of two sets of chromosomes. That is going to be duplicated before we undergo meiosis, which means that we have two copies of each chromosome. We have two copies of each chromosome. We have two copies of chromosome 1 for dad and two copies of chromosomes for mom. That would also mean we have two copies of an X and two copies of a Y chromosome, or 4 copies of X chromosomes. During meiosis 1, and we'll see this in a second, we're really splitting up homologous chromosomes. We're not separating sister chromatids. We're just taking two copies of each chromosome and separating them apart. So this might be mom and mom, dad and dad, or this might be dad and dad, mom and mom, mom and dad, dad and mom. Some variation occurs in which we're separating the duplicated sets of homologous chromosomes. And homologous chromosomes are just the mom and dad versions of a specific chromosome. This gives us two daughter cells that are haploid, but do contain two copies of each chromosome. That brings us into meiosis 2, where we separate single chromosomes, but they're sister chromatids. So meiosis 2 often looks a lot like mitosis. The difference is that we only have half a copy of the total set of chromosomes that we started with. So that will result in anaphase and then telophase, and we end up with 4 chromosomes, or 4 cells, that contain one copy of 1/2 of every chromosomal pair. If we look at that a little bit closer, this is what meiosis 1 looks like. We have 3 pairs of homologous chromosomes. all lined up on the metaphase plate. This is a function that we call independent assortment. We'll talk more about this in future classes, where you can have every pair of homologous chromosomes line up independently of each other. So it's not a middle school dance where all of the boys are on one side and all of the girls are on the other, and there's just some awkward pauses. We tend to see independent assortment occurring between the male and female versions of each chromosome. And then we split them up. So if we look at the result of meiosis 1 into meiosis 2, here are my 3 maternal chromosomes. Here are my 3 paternal chromosomes. Note that 3 refers to the chromosome. P and M refers to dad and mom's copy. We split them up. So these 3 go over here. 1, 2, 3, mom, mom, dad. And then we have 1, 2, 3, dad, dad, mom, and then those sister chromatids will be split up. Now I do recognize in this image that we see a mostly blue chromosome with a little bit of red, and a mostly red chromosome with a little bit of blue. For right now, we're not going to think about what that is showing, but that will be something that we address in future classes. So again, when we think about meiosis, it's 2 flavors. Splitting up cells into 2 cells with 2 copies of half the chromosomes, and then splitting those 2 cells into 4 cells with 1 copy of half of all of the chromosomes. I know that I've just thrown a lot at you, so use the slides, use the video, and come in with questions to start class off tomorrow. Have a great day, and I'll see you soon.